Hi, everyone. Welcome to Friday Machine Learning Barbecue reading group. Today, I thought would be good to go over sort of these new findings by a group over in Washington talking about how the M1 region of our brain might be dual um, a dual system for brain functional regions. So they basically have worked on this for the past 2 to 5 years, depending on who you ask in interviews. The lead researcher, Nico, actually states that the old model is false and wrong, and they're currently trying to be better at using this into a clinical form. But this Nature paper is thick. And so I thought it was very interesting as someone who is looking into a lot of the different brain regions and how to parcellate a brain, the different regions, why you might look at certain brain regions or cortical gyri or you name it. And so this just appealed to me. It is a very thick paper. I did prepare quite a lot, but I probably will miss something as this is a neuroscience heavy rather than computational neuroscience heavy. So if you do find things that I didn't say or discuss or you have questions for, please feel free to interrupt me. This is a very easy presentation, relaxed on a Friday, so just let me know. So to start us off, this paper is coming out of Washington University, published earlier this year. It suggests that there's two intertwining behavioral control systems in the human primary motor cortex, or M1. Specifically, it's made up of effector-specific circuits, really responsible for precision, isolated movements of specific body parts, specifically fingers, toes, and tongue, which then are used for complex tasks like speaking and or manipulating objects. This second network, they call SCAN, or Somato-Cognitive Action System. I'll say that again, SCAN for Somato-Cognitive Action System, is more integrated, involved in whole body control. So this is to think of like integrating motor control, autonomic response, and action planning. They specifically say the scan is comprised of specific regions in M1, the supplementary motor areas, thalamus, posterior putamen, and posterior cerebrum. With all that being said, those are the two regions I'm going to hopefully try to build up to in the two networks that are intertwining with each other to form this idealized version of the homunculus. Great, little background. Everybody, Penfield in our neuroscience classes. They mapped the brain back in the 40s using different types of tasks. Specifically in at least in the 30s and 40s, they mapped out the M1 with cortical stimulation. They found that half of the stimulation sites illicit movement, mostly in the hand, foot, and mouth, leading to this idea of the motor homunculus. Penrose actually didn't develop this schematic himself. It was actually a nurse working in the hospital with him. He had a dream about it and finally asked someone to sketch out what he was thinking. And this ended up becoming the diagram we've all been looking at for 100 years. Basically, studies in non-human primates also show discrepancies with this model. It's really divided into an anterior old M1 and for gross motor movements. So here specifically, we have, or my cursor's over here, in the anterior part of the motor region, we have the old M1, which is what many researchers claim should still have the function of the foot, mouth, or the foot, tongue, and hands. Effectors like the tail and mouth were represented in the anterior M1. They also, findings in sort of these non-human macaque or non-human studies show that concentric zoning of limbs with digits, shoulders, were typically at the periphery of M1. So basically all of this leads to discrepancies with a spatiotopical homunculus model of the M1. So there's some discrepancy. They also specifically talk about how the dorsal anterior cingulate cortex, or the DAA or DACC, and parts of the supplementary motor area are are also indicative of the M1 area. These are the main transmitters of motor commands via the cortical spinal tract, and have implications to the somatosensory cortex, or the S1 cerebrum in the striatum, where really coordination, learning, and inhibition of competing movements are known through different types of effects. Great. So understanding that there are really disparities between what the classical model says of more of a static gross M1 old school model, there is new evidence suggesting that there is more functional connectivity within this, and there are greater tracks to investigate. What do they do then? So they are data driven. They call it precision functional mapping. Specifically, it's what we do within our groups, specifically looking at large sample sizes for effects, especially in different types of regions, causality, different, you name it. So they have so much data. Oh my goodness. I apologize for throwing the paper up, but it was hard to even get into a specific slide. This is the next 5 pages are just describing all the data. They not only have the three large fMRI resting state data with UK Biobank, HCP, and the Washington 120. So they roughly have, I believe, 1,500 resting state scans that they're using. They're also using the CAC data, and they're using infant data, adolescent data, neonate data, and peripheral stroke data. So with that, I believe they're on the order of 10 to 11 data sets that they are using. Each have their own parameters, each have their own mapping in terms of, or functional scans that they use and structural scans that they use. Some of the data sets are using T2 weighted, some of them are using T1 weighted, depending on whether or not they're looking at subcortical features or not. But just rest assured, I think everything here looks okay as going through. One hesitation I had is that they are doing seed-based functional connectivity. This is okay, and I'll show you on the next slide which ones they end up choosing, but I don't typically like function or seed-based functional connectivity, only because I do know that the brain is connected everywhere, all the time, all at once. But regardless of all of that, they do find very parsimonious results within each of these data sets. To find the larger large-scale networks, they use InfoMap, which is a type of sparsification on graph and trying to discern the different sets. Each of the discerned regions are then qualified by a neuroscientist to make sure that they have the correct areas labeled. They're also doing functional connectivity on subcortical regions, and they're looking at the lag structure of the signal to try to discern any kind of ordering of firing of regions. Okay, all that being said, is there any questions that I missed on the data or methods or the way that they set these up? I know I just talked for about 10 minutes about this. Any questions before we get into some of the results?
Did they have different, so they're integrating like 3 or 4 different studies here, right? Did they all have the same PR? Were they all gathered the same as UK Biobank? What was the—
Man, yeah, so without going through the 5 pages of details, no, they were not all gathered very similarly. There were different scanning types, there was different field of views, there was different sequencing patterns, mostly T1 weighted EPI echo based structural imaging, and then MP RAGE resting state functional scan. With that though, I do know that they call it PFM throughout the paper, which I don't normally see, but they're basically claiming that if we can use the 3 largest data sets that are publicly available and use as many participants as possible, that this should be good enough to show a parsimonious effect.
Okay, at least within a few countries. Yeah.
Yes, and include, I mean, limitations on this both include demographics. They don't really show demographics on ethnicity, age, or they show some age, but mostly this is 23 years and younger. So
Right.
no one over any kind of cognitive deficiency or atrophy that happens just with aging. Yeah.
Or where's the Wu data set gathered? Because I know UK Biobank, I know HCP, but
Yeah, Washington University 120.
that's Washington.
So
Okay,
yeah, I apologize. I just used abbreviation there.
it should have been clear.
Yes, I believe they also do with the Washington University. They do anterior to posterior scans, and then they do posterior to anterior scans as well to try to match structure, just to make sure that there's fidelity between the scans.
Okay, interesting.
Okay.
Definitely demographically for making these kind of claims, but one study.
Yeah. Yeah. I mean, gosh, I wish we had an HCP for like each continent. That would be great.
Right.
But we don't. So unfortunately, we gotta maybe we spend the next $10 million on that.
Yep.
Great. So this is the beginning of the first set of results. How do they look at all this data? Specifically, they are interested in the M1, which has many different subregions. Specifically, they chose 6 different seed regions as outlined. Are you all able to see my mouse? Yes. Okay. Kind of.
Yeah.
Okay, so here in A, the 1 through 6 here are all the seed regions that they describe within the paper. This is their precisional functional mapping. So this is locations in the left precentral gyrus in one of the participants. There is a supplementary video I did not get to that shows sort of the activation over a time series. But they have one being is more activation when the foot. They have 3 saying that that is the highest correlation when the mouth is activated, and 5 is the highest location when the tongue is in motion. As well as integrating of strong interconnected regions. 2, 4, and 6 were also very high in correlation when doing the seed-based analysis. One thing to note is that they, for B, here we have the individual, just a single person with the seed regions. These are the task invoked regions that are shown, so foot, hand, mouth, and then these are the group averages. There is strong correlation here, so they felt like these 3 or these 6 seed regions are okay moving forward in terms of analysis. For figure 2, these are the functional connectivity and cortical thickness of the M1 inter-effector motif. So in the first, when they do the seed region analysis, they are looking sort of at what is happening in the interaction between many of these regions. One thing that they started to describe and use as a definition is they called it the M1 inter-effector motif. And specifically here, they found 2 distinct patterns of functional connectivity, differing from the traditional understanding of M1. The first pattern that they see here is the foot, hand, and mouth, which each region is a homeotopic contralateral M1. So all that means is just like here, everything is happening down the M1 passageway within the brain. And so what they're doing then is they match activity during the foot, hand, and tongue movements. There seems to be an existence of this inter-effector region when they looked at the different data sets, and their second pattern emerged where the 3 regions known for the foot, hand, and mouth. These regions form in. Yeah, they perform they form an interdiagonalized chain down the pre-central drivers, previously unrecognized and termed the inter-effective regions. So just going back, I apologize, I wanted to make sure I get my notes. This strip right here, what they're talking about, is the inter-effector region of M1. This is where most of the activity is being popped up using the seed-based functional activity. So these were also verified in individuals across stages of development. They had an 11-month-old infant, and they also had a 9-year-old child that specifically showed that they both correlate with the same findings as the adults and showed that they have the same activity and spatiotemporal pattern. Even though, yeah, I even have notes that they even showed these types of networks within bilateral prenatal stroke victims, damaging large portions of the M1 in demonstrating that there is this interaction between networks. So what does this mean? So they talk about this inter-effector link to control networks. So for here, this is the brain, for A, these are the brain regions with the strongest functional connectivity to the middle inter-effector region. So this is a part of one of the seeds. So we have the supplementary motor area and then the dorsal, or the DACC, the cingulate cortex. But they all pop up when using seed-based correlation when starting at the cortex, striatum, and thalamus, and the cerebrum. So all 4 of those seed regions do end up highlighting these 2 specific regions. And then their correlation within the striatum, cerebrum, and the thalamus. For B, these are the regions that are most correlated when the foot, hand, and mouth regions are used during tasks. Purple here outlines the cingulate occipital network, or the CON network is what they describe. So effectively executive control. There's so many names that people like to give large scale networks, but this is essentially executive control for fine motor movements. And specifically, they call this CON, or the cingulate occipital network. And so the central sulcus is masked and exhibits large differences in definition for this region. They talk about the functional connectivity within these regions using the seeds on these 3 tasks, how the CON has a functional connectivity higher than most other defined regions, all of which they had to hand choose where they were based off of the neuroscientist definitions after using InfoMap. But with C, I apologize. For C, I'm not a huge fan. This is a spring embedding plot. Does anybody, have everybody ever used a spring embedding plot before?
Embedding, right?
Yeah, it's concatenating all the regions, and then they're judged on the disconnected parts and pushed away spatially as a visualization. What this is trying to say is that the CON network is then leading to the inter-effective network, which goes to hand, foot, and mouth in some sort of causality. They're trying to find a temporal or a downstream ordering of operations for the pathway. So same here with D, the temporal ordering of these. This is specifically systematic differences in the temporal ordering. This was done in the infraslow signal, so 0.01 to 0.1 hertz. What is interesting about this? Prior electrophysiological work suggests that the latter infraslow activity corresponds to earlier delta bands. So here what they're saying is previous work has been shown that there's a temporal ordering, but it might better correspond with higher frequencies. they suggest that further study should be done, especially with multimodal data. E, however, for each participant, the inner effector regions exhibited lower cortical thickness than all effector-specific regions. So the inter-effector regions just have lower cortical thickness, so there's less neural packing within those regions. I am not sure what that means, but it is important, I think, for attention, memory, and potentially integration tasks. But that's just to be of note that they also were looking at cortical thickness regions within the effects of the specific regions that they were looking at. We then get to tasks. With tasks, they try to understand the inter-effector regions in the primary cortex using the tasks. They draw, the data was collected from 2 participants doing 25 different movements in novel tasks involving separate planning and execution phase for coordination of your hand and foot movement. So specifically, they were looking at all of these tasks separately together in trying to understand how their findings both corroborate and disprove many of the traditional findings. So what it suggests is that the organization of the primary motor cortex is more consistent with a concentric model rather than the traditional homuncular model. This centric organization this organization has zones of activation centered around activation peaks for distal movements, such as if I'm moving my hand, my toes, my tongue, and then expanding outward more proximal movements, such as shoulders, the jaw, any kind of head movement. And so as you would see here, this is the participation 1 and 2, the movement map. As you go more frontal, my mouse is over here. As you go more frontal, ventral to dorsal, parietal, you're going to have more effective contralateral movements. So as you see here, the movements here are very nicely color coded. You get tongue, swallow, jaw, nostrils, eyebrows. So a lot of the ways that you go from frontal to parietal is going to be smaller movements to larger coordination, is what they are talking about. Here, B is saying activation strengths for each movement was computed along a dorsal ventral axis. And so a 2-peak Gaussian curve was fitted to each movement activation. Fitted curve shows for movements of abdominal, shoulders, elbows, wrists, and hands. And so this is just showing that as you go from more ventral to dorsal, these peak activations really occur in specific areas. Great. C is the co-activation during abdominal contraction. So there's more coordination between many of these specified movements predefined based on larger coordination. So abdominal flexation, movement specification. D, we have the evoked activity during movements was computed as the 6 conditions most activated each discrete region. So the 6 movements that specifically activated each of these regions were toes, abdominal, hand, eyelid, tongue, and swallowing. And then there are event-related tasks that are also adjacent to some of these predefined regions, where the event-related action plans were separated for different movements. And this is further in the supplementary section, which I did not get to, so I apologize. But all this is saying really is that they want to go away from this concentric model of the homunculus to more of a dual system where you have both the concentric in effector region where there is more fine motor movement displayed depending on your region, and then higher level coordination where you have more action body, action body, action body as you get to the shoulders or higher wrists. So it's more of a continuum. They suggest this is a fun depiction, but should not be used as set in stone. As many times this should just be used for medical students to learn the functional the functional anatomy of the brain. Specifically, yeah, that's what I want to say about that. In terms of other studies, where are we at in time? 30 minutes. Yeah, in terms of other studies, I can go into the macaque homunculi of the action body network. There is effector isolation. So meaning, how do they look at the different regions to suggest this new physiological stance, and then they have a bunch of passages to suggest evidence towards electrophysical or electrophysiological evidence. They have evidence from clinical neurobiology or clinical neurology and similarities to sensory systems. So they suggest this is a network for mind-body integration. Yes, I have I have about 8 pages of notes, but I'm not going to read them to you. So basically, the takeaways of this paper and what they really strongly suggest is that we discard the idea of the homunculus altogether. It's great for historical purposes and use for larger scale history of neuroscience, but we should move away from this and think about more functional somatotopy organizations of the brain. These interventions exhibit really decrease in these networks, and so there is a structure of there's the structure of these two networks that intertwine together, doing both fine motor movements and action planning. This was confirmed by 3 of the largest data sets: monkeys, pediatric cross-species, homologous, and precursors to the effector system. Last things are range of actions showed that they can separate by these con network linked inter-effector regions. They were activated by the action planning and axial body movements. And then combining these findings with other studies suggests that this M1 has this Somato-Cognitive Action Network scan in conjunction with the inter-effector network. So with these two parallel systems, the effector regions are isolating the fine motor movement scan, integrating goals, physiological or physiology, and body movement. That is what I have prepared for you. I have a bunch of notes I'm happy to share with the chat as well if you're interested in going through some of the deeper sections. But yeah, I guess what are the implications of all of this? I was listening to a study I was listening to a podcast with the original author on it, and they are particularly interested in applying this more towards people with different types of motor deficiencies, sort of people who have loss of limb, people who are paraplegics, people who have Parkinson's or different type of motor deficiencies. And so looking at sort of the abnormalities with this new framework is sort of the direction that they want to go.
If we have any like historical lesions in these inter-effector regions that are suddenly like, oh, this like that makes a lot of sense that you know we've seen lesions in these regions that didn't you know, sort of line up with the homunculus before, or that maybe we can with this idea of the inter-effectors were given new insight.
Yeah, so they did talk about how there are probably planning areas, especially since many of what they're finding is more high-level action planning. We introduce like proprioception or stress response, specifically even when we're thinking about planning. And what they've said in part of one of the novel tasks that they were doing is this idea of planning to execution. And during the planning phase, you can see various differences between the two networks and how they are interacting with each other, especially because one network will be on, meaning the planning, meaning the action body one, whereas the effector network is off. Does that make sense?
Yeah. Yeah, I'm curious to see, you know, as it's only came out in April, so there haven't been any big replications yet, but it'd be really cool to see more replication studies. And again, like I'm interested in lesions in these areas in particular and how that would affect.
They did have
such an amorphous thing to me, right? And how it would be affected by neurodegeneration, I think is interesting.
Yeah, one thing that I hesitate with this study, and I know it's probably mostly with all studies, is I hesitate with anything subcortical. I know that they have plenty of information on how they captured T2 weighted images to better have precision on the structural or the subcortical structures. But when they're looking at thalamus, cerebellum, different types of brainstem regions, I'm just always so hesitant.
Right.
So I know that they were using those as even seeds, and I know they have 15,000 participants or scans that they're going through. So maybe my hesitation should be quelched, but I don't know.
Well, I think it's promising. I think yeah, it just needs replication, and like I think we'll need to look at with some other. Like I'm also skeptical of seed-based connectivity, so I'd be interested to see if these regions pop up again with a non-seed-based model, even if they're somewhat like washed in signal, which can happen when you don't use the seed-based method. But you would expect them to at least pop up somewhere.
Yeah.
Yes, I think that's going to be a cool thing to look out for us. Future work kind of backs this up, or even retrospective work. I mean, you don't have to do new studies. You have this new way of looking at how the brain is, the cortex is divided up. You can test it on old work and see if it lines up with old hypotheses, or if it like just completely flies in the face of everything. Then maybe
because one thing they were saying is that with the they have different pathways that they were talking about as well, including ones that include both the motor cortex and the somatosensory cortex, talking about differences in proprioception, pathways. They said that they would like to continue to look at the somatosensory area, but it probably may just be a rabbit hole that they go through, only because they were finding effects with it. I do agree, seed-based. It's just very. It's just one way of looking at it.
Yeah, it's just one way. I think that hopefully it wouldn't constrain their analysis too much. I think again, another analysis pipeline would show some similar conclusions. That's going to be the test of replication and the test of model bias.
Yeah. I do really like, though, that they did do a full development and cross species. I'd be curious if this couldn't be done in rats, because I don't think the motor cortex is well developed, or it just has different features. This is like barrel cortex, like we don't have. But I don't know. I'm trying to think if there's another. Does anyone know if pigs have effective motor regions that are as developed? Because they were talking about evolutionarily, there's a part, I think the preputamen in macaques are less developed or more newly developed in structure in terms of evolutionarily. And so that was one of the significant differences between humans and macaques. And so they did find some differences sort of around like jaw, larynx, eyes in those areas is what they were saying. But I don't know. But that is, yeah. I know we have 20 minutes. Yeah, go ahead.
Sort of, sorry, how it's a little bit odd to me that there's only like 3 of these like interconnector regions. I would expect there to be, just like thinking about it, it makes more sense to me that there would be more. Like, I don't know. Like,
yes.
That makes some sense. Like there would be more, and there would be more like as you have the verdigris, that there would be more, and they would appear more frequently between like especially like different kinds of motor movement, different limbs. I don't know if that makes any sense.
It does, and I think it's just because the way that they define their cingulate occipital network, the con for executive control, and that's really what they were looking at. So really, they were just kind of in these regions, in motor specific really did light up these 4 gradient really of regions. Whereas then when they started to do action planning and more of the more complex tasks, they got very isolated regions or areas of activity, which is why that they were then starting to say this. They also say this is a single slice on the brain and they don't show a gradient over more of a 3D topology. But yeah, I agree with you.
Yeah, I'll be curious to see. I think it's a really cool, finding though initially, and I think it'll just get refined with further work, hopefully.
I also think it's just funny that this is their idea of a tongue. These are the 2 eyes, and this is sort of what speech integrates between the 2.
Yeah, I was wondering what that was. Yeah, it seems like it really is in need of like some kind of corroboration with lesion studies.
Yeah.
Because I think the original homunculus, as imprecise and probably flawed if it is, was somewhat supported by neuropsychological studies involving like double associations. So it'll be interesting to see whether or not they can kind of confirm this model behaviorally.
Yeah, that's where I was like wondering. I was like, planning is kind of a hard thing to.
Yeah. And some of these could be correlated, but the correlations could be epiphenomenal. Right. You could have lesions that are just totally unrelated to behavior.
I agree. Yeah, do we know do we have access to any lesion data sets? Are there open source large scale lesion data sets?
There's a ton of different lesion sets. I know a few white matter ones. I don't know about cortical lesions so much, but I'm sure if you look around, you could probably find some, even some modern ones.
Yeah, I just didn't know if anybody knew of anything off the top of their head for N of like 50 plus. I know there's always the 10 or below or like a specific studies looking at lesions, but without having to conglomerate them all or concatenate them all. I was just wondering if anybody knew anything of that. If there's anything out there even exists like that.
Nothing cortical offhand. There's the Valdo competition data set is a nice white matter data set. I think that skews slightly older, but it's all white matter. Well, I guess they do microbleeds somewhere on the cortex.
Okay.
But yeah, it's also, I don't think they have any tasks for that competition.
Yeah.
And people have done some lesion discovery on UK Biobank actually. So I wonder.
Oh, interesting.
Alignment there. I'd have to look. That's a good question.
Yeah. But anyway, this is really what I prepared. I'm sorry I didn't go to the hour. There's just a lot to this paper that I didn't get to, so I apologize. But hopefully this is a good introduction. Are there any questions about this? Any other hesitation, thoughts, comments? How might we apply this? I'm happy with the discussion.
I mean, it would be just interesting to take a non-seed-based connectivity, approach and see if you can just plug it into their data. At least one of the data sets we have ready access to, so you can get started on UK Biobank.
So, if anybody knows this, but Dr. Calhoun was telling me that he and Benham were working on a deep learning project where a network very similar to this popped up in their results, but they didn't, I don't know if they knew what to make of it. And then they saw this later.
Oh, nice. Well, that's cool. That means that it would be worth publishing even just a blog post as a corroboration.
Cool.
So they were finding planning networks, is what they said?
No, I think that they were just analyzing motor regions. And this popped up as kind of an unexpected result that the model gave them. So they weren't sure if it was, because I don't know, because it wasn't something that they were looking for. And then based on prior work, I think maybe they might have suspected it was noise or something.
Were they looking at resting state though? That's interesting.
I'm not sure.
Yeah. That would be.
Yeah, because what might be interesting is if you do have someone doing more motor tasks or like fine motor movements or the difference between the tasks or however they found the two, it just seems like whenever you're doing higher level planning or more complex tasks, the regions just get smaller. It's just not widespread. And I know that might be more consistent, but I just find that to be I don't know. I hesitate a little bit just because of how small these regions look in
Yeah,
coordination.
yeah, it's a little sus.
So the ones on the very right, on the bottom plots, are those the ones you're talking about?
Yes, so specifically what I'm looking at, they define the different movements regions here. So we have all of these different types of movements. They talked about how from dorsal to or dorsal to ventral that they have different spikes and peaks onto where they are effectively firing. And so they outline those regions again in find. So this task was abdominal flexation, which is a harder task and more coordination. So the effective areas are smaller. This is a movement specific, I believe. It kind of looks like versus, or this one was 6 different movements that they all mapped together, showing that all of these regions are very particular into the 6 different movements I listed before. I can look later, but the last one is the action planning. This is the actor planning and then execute stage, and all of these activation areas are just so much smaller, and so that's why I hesitate.
Scale is also half of the scale on the other 2 plots, which
Yes.
I know scale is not always meaningful here, but it just to me, if I were looking at this and I was like, okay, what is probably noise? I would say the right hand, like those small little dots that are appearing, especially so more like noise, especially the noise that would be cut off by the thresholds in the other two figures. So
yeah. Really cool thing about this though is that it is non-invasive. So nobody had to have electrodes stuck in their brain, which is outstanding. I think it's a cool finding regardless.
Oh yeah, you know, I
still agree. They could definitely do something related to TMS with this. To play around with it a little bit.
Yeah, I think this is like a cool, even though it's such a big study, I would still call it like a pilot method at least, like just because
Basically, it did feel like it was 3 papers rolled into one because they were proving that there's the separation of networks, then they were doing development, and then they were doing separation of evolutionary differences between humans and macaques. So it was just a lot to piece through, and it's a great first study.
Cool.
Yeah, I think, I don't know, you guys want to end a couple minutes early, give you guys a little extra time on Friday?
Yeah, I think I'll definitely keep an eye out. Definitely let us know if you see any replication studies or people using this. Be really interesting to follow up and see what happens with this work.
Yeah, of course. Yeah, I think it should be pretty cool the next year, what we find and what we see. But I'll go ahead and post my notes and the slides to the channel in case you want to go into some of the or my summary of some of the different studies. But yeah, I really appreciate all the questions and back and forth. Thanks, you guys.
Yeah, thanks, TJ. That was awesome.
Yeah.
Okay.
See everyone.
See it. It just noticed that the owl was still on under the one on the TV. I was wondering where the center one was, and I thought we might be back to